Okay, bioinformatics is the area, the thing you can use the computer technology to analyze the any biological information. So that means it's a huge. It is not a one area. It is a so many areas you can consider the bioinformatics role. So that's why it is not very well defined one kind of topic. Some of the topics are entirely not related. So in order to make you a clear, a rough idea what you are going to learn, I have prepared a slide for you. So before that, so next generation sequencing, why we are including this? Most of the laboratories now, they are working on next generation sequencing. It is almost like a must now. Many of the areas research is metagenomics or transcriptomics, metatranscriptomics, or just genomics. Many areas, many discoveries last few years because of this next generation sequencing. It was possible. So though it was started just normal from Sanger's, uh, but advancements you can see from the Roche, that is pyrosequencing, which you have learned in last class. And the next one is Illumina. This is another breakthrough. So this your course just to for make you to understand, visualize the all the areas and topics what we are going to learn. So first we started with the introduction where we gave I taught you some of the applications of it and history of it. That is a separate. Okay. Now we are in the sequencing technologies. As I mentioned to you, sequencing is one of the major area of bioinformatics become, not before. Before is a small part of it, but now sequencing is the main, because the most of the labs are working on the sequencing technologies, and sequencing data is a very huge amount of data you can see in the NCBI and other databases. So it is a must to understand the sequencing technology. That's why we included. We started with the Sangers, and then pyrosequencing, and now we are in the Illumina. And practical wise, Sangers is okay, and is the one only we are going to learn in bioinformatics course, because Pyro and Illumina, if you want to understand that itself, it takes a lot of time and a lot of practical classes you need, and whole course it may require. That's why now we do have the next generation sequencing as a separate course in bioinformatics program. I think some of you will be going to learn in the next year bioinformatics program students, but next generation sequencing as a separate course, where you will learn all the genomic analysis, transcriptome analysis, meta-transcriptome, metagenomics, all those things you will understand during that course. But as a biotechnology students, you must know what is this, the basics of the sequencing. As any lab, if you go, that is going to be, we are going to work on the sequencing technology. And in fact, actually, this Illumina started, a couple of years back, 2011, when it is the first mission they bought in UITM. They conducted a workshop to show how it works and what it is. And I attended that, and I found it is going to be a rule. The thing is, I found that time itself it is going to be a big milestone in the sequencing technology. And immediately included in our syllabus. Some 2012 onwards, we are teaching this Illumina sequencing technology. Of course, now everyone is teaching, but at the time itself we started. That is one of the example that how workshops can help us to improvise our uh, teaching and our syllabus. So then what we are going to learn is the next topics are core bioinformatic methods. That mean biological databases. One of the class I gave you a brief recall of your understanding of basics of the molecular biology. So because without understanding those things, when we talk about sequences and the alignment of those and translating into the amino acid sequences, you may not understand. Basics are very, very important. Even the sequencing technologies before that, one of the basic I told you, how the nucleotides looks like, and where the phosphodiester bond forms. Without that understanding, you cannot understand dideoxy method, isn't it? So basics are very important. So that's why some of the basics I taught you in one of the class for which are needed for this. Um, and next, you are going to understand some of the databases and sequencing, different sequencing formats, and how to retrieve those sequences from the databases, and then pairwise alignment methods, multiple alignment methods, and phylogenetic analysis. So with this another area. So sometimes it is not much related one to one. So that's why I just want to show what you are going to learn in a area wise. Then and in this you are going to learn every topic. You will be having a practical: biological databases, sequence formats and retrieving, pairwise alignment, multiple alignment, phylogenetic, everything, every topic you have practical. So one way is good. What you are learning you will understand, you will implement it for purpose. And the next one is what you are going to understand is the tools for in silico cloning. So most of you who learned the genetic engineering, you are understanding what is meant by cloning a gene of interest and putting into the a vector and get a recombinant plasmid. For that, you have learned what is the restriction enzyme, but we can use the some of the programs to analyze the sequence to understand what are the restriction enzyme sites are present, etc. So that tools, this is comparatively very simple and tools which you are going to learn. And ORF finder. So the based on the sequence, unknown sequence or your sequence, gene of interest. So ORFs we will find a, and primer designing. So primers also, which is needed for the PCR center, etc. And manually we can do it by knowing the what and some of the properties, but online tools also there to design the primer. So that is also you are going to learn. That is one of the area. And uh, and here also you will be having practical for everything: restriction enzyme analysis, water finder, and primer design. And finally, you are going to learn about comparative genomics, where just theoretically we will teach you. There is no practical component of it in this, because comparative genomics more detail you will learn in as one of the elective course in final year, and including biotechnology and bioinformatics. Both students you will be having an option, but biotechnology is an option as a comparative genomics as a elective, but bioinformatics students will be having a core course on comparative genomics. So this is the whole thing about rough idea of your bioinformatics course, what you are going we are learning. And this part is finished introduction, and now we are in the sequencing technologies, where we finish Sanger's and practical also. I hope you have done it. Maybe we can see later whether if you have any queries regarding it. And Pyro we have learned, and Illumina today we are going to see. So with this, sequencing technologies will complete, and there then we will be entering into the core bioinformatics methods, where all your textbooks you will be having these things, only these things most of the times. And then yeah, fine. And practicals, right? So all these practicals, one of the um, uh, a good thing about it is you will be using these practicals not in the form we are going to assess, not in the form of practical reports, not in the form of practical examination, but practicals here we are going to use for your project. So uh, we will be giving you a small project for you in silico project, where you will be going to learn. You are going to use all these practicals for your project. So that is how it will help you to not only just understanding and that particular class. You are going to implement for for your project. So that is one of the beauty of it, and easily can be done. So not very complex things to understand. So utilize also, and we are going to assist you for the your practical, all these practicals in the form of practical report. Sorry, the project report and the project presentation. So thus, there is a high chance you will get all of you A in this course. Unlike last year, many of you lost some of the marks in ASPE examination. So, um, and yeah, I think we couldn't help because I didn't ask you much questions also in ASPE. I don't know how you perform, but I can see many of you lost the marks in ASPE. But here, all the practicals is no ASPE or no practical reports. So mainly by project presentation and reports. So I think everyone will do best, I think, and you will get A in all of you. So this is about the once again, to have a mind mapping what you are going to learn or what you are learning. So now we will continue with the Illumina sequencing. So the learning objectives, usually I don't put this one separately, but I just explained to you, but I put here. So what you are going to learn is a principle and procedure of the Illumina sequencing you will understand, and what are the applications. Once you understand the output of it, you will understand automatically the what are the applications will be, and you will understand and appreciate actually this NGS role in the deluge of your sequencing data at present. Um, you know, yeah. So this is the Illumina sequencing workflow. This required 4 steps. Okay. So one is a sample preparation, the first one. Second one is a cluster generation. Third one is a sequencing by synthesis. And fourth one is pipeline analysis. Okay. So we'll try to understand step by step, one after one. And if you have any queries, you can ask at the in between. I'll try to clarify. I'll try to explain in a different way. So always, face to face, it was very easy. I can see where you're lagging, maybe by asking the questions, and also I can see your facial feelings. I can see where you are having some issue or problem in understanding any step, so that I can repeat in a different way. Sometimes I can tell the same thing in 3 or 4 different ways so that you can understand. But now it is not interactive way. I cannot see you and just keep going, just telling, and I don't know whether you are understanding or not. And please let me know in any step you are not understanding, or maybe at the end also you can explain, ask me where you are not clear about it. So uh, among these four, so first let us understand the step, each step by step, and let us stitch together to get overall understanding of the flow. So the first one is samples preparation. So where you can see the sample, that means DNA or RNA. So but RNA, when you want to sequence, you have to make it into cDNA. So reverse transcribe it and becoming making into DNA. So thus you can sequence RNA or DNA. That means you can sequence genome or transcriptome. Okay, so DNA, you are making this way sample preparation. You will add some of the adapters, which is you can see in the red and blue colors. What are those? Those are the adapters you added artificially. Okay, so then in this step, first step. Second step, it is called as cluster or cluster generation, where you will make each one into a cluster like this, and the third one is a sequencing. So um, this we are going to learn, and where we do it. So how we do it. First one is a manual one, so no need of any machine. So we no need of any robot. We are going to use tubes and tips, and we are doing on the workbench, and that requires different steps. And second one, automated cluster generation. Cluster generation is an automated mission. So we just give all the ingredients which are needed for it. So it will generate all the clusters. So these are the different steps in this particular step. And the third one is a sequencing. Again, it is automated. This is a main sequencing mission, which cost a lot, and this also having different sub steps for this. Okay, so this is the first one, which is library preparation, which is taking 6 hours of time. This is a 5 hours of time. This is around 3 days. So by this another almost like a 3 to 4 days, you will get GB of data. Can you imagine? You used to take years of time, just 1 or 2 decades back using the Sangers, but now you will get a GB of data just in a matter of 4 days, and just one person is enough to do, and one mission is enough to do. So that is the very big breakthrough in the sequencing technologies. Okay, let us understand step by step. The first one is a library preparation. So what we are going to do in this? So we are going to make the DNA into fragments, just like your pyrosequencing. Here also we are breaking the DNA into fragments. So those fragmenting can be done in mechanically or enzymatically. There are some enzymes where it will bind to the DNA and roll on it, and every certain intermediate of the length it will cut the DNA like 100 or 300 like that. So that fragmentation can need to be done at the beginning, first one. And also, there are so many enzymes are present. Genetic engineering, you have seen some of the enzymes, and where it is actually, it's most of the enzymes is and is naturally available in a different organism for different purposes. Nature naturally evolved, but we are using those enzymes for our genetic engineering purpose, based on those property. That is how we are doing this fragmentation of the DNA also. And repairing the ends of the overhanging. So when you cut the DNA, sometimes you will be having overhangs. That means two strands maybe exactly it may not cut. So some of the DNA, one of the strand will be extra, will be there, few nucleotide extra, which is not having a pair, either 5 prime or 3 prime. So that is called as overhangings, that you are understood, right? So those overhangs need to be repaired. So need to be first fulfilled, and make it two strands equal there, two sides, both ends. So that need to be done. Then after that, add a overhang. So you need to add extra A's both sides. Just add A using again some enzymes. What is the enzyme required? Poly A polymerase. So, so there are so many enzymes are there. Okay. So poly A polymerase. What is the function of poly A polymerase? It will just add A to one of the strand of the DNA. Okay. So that is how you will add it. Then what is the next step? Ligate the adapters. So the DNA, until now what we are talking, we don't know the sequence of it. Without knowing the sequence, what we have done, repairing, that means both strands to be equally present. Another thing, without knowing the sequence, what we have done, adding the poly A tail, this side, that this side. Okay, 5 prime and 3 prime. The next one is ligating the adapters. So the adapter is nothing but a DNA sequence, which you know the sequence, you design those sequence. So that small adapter, maybe around 50 nucleotides length something. So that adapter you are going to attach to the 5 prime. One adapter at the 5 prime, another adapter at the 3 prime. So again I repeat, adapter here is nothing but a DNA sequence. So you synthesized in a laboratory known sequence. You know exactly what is that sequence. First nucleotide to last nucleotide, you know the sequence of it, and that sequence you synthesize. There are so many synthetic methods, commercial way. Even now also we can order. If you want to know some sequence, you can just put what you sequence you want. You can order it. So that is how we get the primers for PCR and sequencings. So that's those sequences. What you are going to do? Add it to the 5 prime, one adapter, another adapter at the 3 prime. So that is what you are doing in the library preparation. So with that, thus the library preparation step will complete, which required approximately 6 hours. But the very good hands-on skill is needed to making this library preparation. Any wrong in this library preparation step, whole sequencing will get wrong, and you will not get the sequencing result. Okay, so in order to not spoil it, so need to be very accurate, very efficiently need to be prepared this library preparation. I hope you understand until this library preparation, and let us go to the next step, which is sequencing. So this one, why included? So library preparation of the things, it's not only for the genomic DNA. So you can take the sample from genomic DNA, small RNA or mRNA. Chromatin binding DNA for the chip sequencing. There are many other different samples, different kinds of DNAs, different kinds of RNA. But DNA just directly breaking down into small pieces. RNA, one step extra, you need to reverse transcribe into cDNA. Then again with the same steps, the breaking down into small pieces, then ligating the adapters. So the second step is automated cluster generation. So as I mentioned, it is automated mission will be there. So it will generate the clusters. So this is a very crucial. If you understand this, the next step also is uh, easily understandable. But this is the thing you understand, and you can appreciate the way how the scientists who developed got the idea to make the parallel sequencing and getting the GB of data is possible. So here we have three steps: hybridize to the workflow cell, extend the hybridized oligos, and perform the breed amplification. So more than that, it is sub steps are there. We will see one by one. So for this one, we are working on this flow cell. You can see there's a small chip which is present here. This is called as a flow cell. This flow cell we are using here for the cluster generation. This cluster generation library turns libraries into clonal clusters. So each library at the beginning we have, so DNA library we have, each one having different sequence. So those we are converting into clusters. So each DNA we are converting into clusters, clonal clusters. And you understand what is meant by clone, right? Clone is the meaning you are making a same into multiple copies. That is a clone. That is you can convey the same meaning into different way, different places. So for example, in the genetic engineering, you have cloned. That means the recombinant plasmid you are making into multiple copies, same copy. That's why we call as the cloning. So that is how the word comes, gene cloning. So that means a gene of your interest put into a plasmid and making in that copy into multiple copies. From a single copy you are making into multiple copies, that is called as a cloning. So gene cloning we call as here. So the library which is the DNA sequences which we don't know, in each individual sequence we are making into clones. So we are forming a cluster, that's why we call as clonal clusters, which we are going to do on the flow cell. If I show you, you will understand better than reading here. So this is the mission, how it looks like, Seaboat hardware. In this cluster generation, these are the sub steps. So in the second step, what I mentioned, the cluster generation, right? So in this cluster generation, you do have sub steps, template hybridization and initiation of the extension, then amplification, then linearization, then blocking, and then primer hybridization. Okay. So before that, I just to need to inform you. I think all of you must be understand before PCR, right? PCR is what? Um, I just repeat the PCR steps once again. Just try to listen. PCR, to recapping, PCR, the DNA you have, the first step what you do, denaturation. So that means double stranded DNA you are making into single strand. Okay. So how you are doing this? In case of PCR, you are by increasing the temperature. Okay. So by increasing the temperature, you are disturbing the hydrogen bonds between the two DNA strands, so that it will be denature and present as a single stranded DNA. So then what you're going to do, you are adding some primer sequences which are complementary to the one of the strand, and you are making the both to get annealed, because complementary, it go and bind automatically. So in order to go and bind automatically, so the elevated temperature 96 degrees or 100 degrees is not feasible, so it will still present as a single strand. So you are lowering the temperature slowly, you are lowering the temperature around 50 or 58 or 60. It will go anneal each other based on the sequence. So each sequence required different temperature of annealing temperature. Okay. So it will go anneal. Anneal in the sense it will go and bind complementary to each other. So the next one is what is going to be happen. You are going to amplify it. So the DNA, one strand is there, another strand is complementary bound, and then DNA polymerase also present in the reaction mixture, and D-entropy is also present. The next step is just elongating the primer DNA until the end by using the template DNA as a message. So it will make the 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 second strand synthesis. So for that you need another temperature, so which is feasible for the DNA polymerase which you added. So most of the PCR what you are going to add is a tag polymerase. So for that 72 degrees is the ideal temperature for elongation. So 72 degrees you increase the temperature, so it will just get elongated. Understand? So you understand why we are changing the temperatures. Okay. So why we are putting the 90 plus temperature, why we are putting the 50 or 60 degrees for the annealing, and why 72. So after that, you will repeat the same things to get repeating the same things and make multiple copies of the same DNA. So once you understand that, this is understanding will be much easier. Okay. What is the thing happening in these steps? We will understand by the slides, which you can see. The flow cell is, this is kind of thing which is the chip which you are putting into the this mission, the cluster generated mission, a small one which contains around 8 channels. 8 channels, you can see one here, the columns, all this, each column is a channel, those channels, and here the small holes are there. So, and here another holes. So each column or each channel is individual, is not related to each other. So this small hole, you can introduce some of the ingredients to there. A small hole will be there, and it will go enter into the channel and flow through here, and it will come out from here. Okay, so that is how it was actually manufactured. And there are 8 channels are there. Again, I repeat, each channel are independent. Nothing. So once you any reagent you introduce to the first channel, so it is nothing to do with the second to until 8. So only it will enter into the first channel and flow through here and come out to the from the first channel only. So so that is the flow cell, how it looks like. And inside, if you zoom it in the microscopically, electron microscopically, because you will not see naked with your naked eye, you can see these kind of primers immobilizely present on in the flow cell, each channel. So millions of it, not a few thousands or few millions of it. It will be present as a lawn. As a lawn, it is present. Okay. So here, what are these primers? Primers is nothing but a DNA sequence, single-stranded DNA sequence, which is your known sequence. And here, another thing I need to mention here, there are two kinds of primers, not all same. So you can see the colors difference. One is in orange color, another one is green color, another orange green. So orange whole line will be orange, another line is green, another orange, another green, another orange and green. So thus whole flow cell, all the channels are occupied with a lawn of primers, the two sets of primers, which are at present, just I will explain to you in the color wise, orange and green. So what these two primers, two primers, if you still remember, in the first step, what we have done, the DNA is we make it into small pieces and we added the adapters, 5' one adapter and 3' one adapter we added. So these two primers are nothing but complementary sequence to these two adapters. So one of it, the color wise, just to illustrate only, uh, to just give an example, say let's say orange color is the complementary to the adapter which you added to the 5' side, and green color is, let's say, complement to the adapter which you added to the 3' side. Okay, so that is how it is, the design is. Okay, the flow cell is present. Okay, so surface of the flow cell coated with a lawn of primer. So these are, you can see the bottom of it, it is fully covalently bound, is immobilizedly bound. Okay, so you cannot remove these primers from the flow cell. It is strongly attached to there, so it will be present there. So that is how the flow cell looks like, and the primers are distributed on the flow cell. So the next step is what? The double-stranded DNA with, okay, then what we are going to do, the flow cell, right? So the hole present here, it will be connected to the some channels in the automated mission, where we are going to introduce your the DNA product which you got in the first step, that is the library of the DNA. So DNA library we are going to introduce to this flow cell. Okay, Those library is what? It is having a double-stranded DNA, which is having 5' one adapter, 3' one adapter, and the DNA sequence which you don't know. So once you introduce that, so that DNA you are going to denature by sodium hydroxide before introducing into the flow cell. Okay, so the library preparation, what we got was double-stranded DNA, and which is having the 5' and 3' both adapters we do have. And those double-stranded DNA, before introducing to the flow cell, we are going to denature. How to denature? You try to understand in the PCR, we denature by using the elevated temperature to 96 degrees. But need not be only the way for denaturing. So the denaturing is nothing but you are disturbing the DNA, the hydrogen bonds between the two strands. So one of the way only the heat, elevated temperature. There are different ways are there. Some of the chemicals also denature the double-stranded DNA. Here we are using the chemical. So one of the chemical is sodium hydroxide can denature the double-stranded DNA into single-stranded DNA, and you inject that sample through the flow cell. And what happens? See, this single-stranded DNA will going to hybridize to the primer lawn. So why it is going to be hybridized or anneal? If you want to use the same term as PCR, it is going to anneal. So why PCR in the PCR, the DNA going to anneal to the primer? Because of the complementarity, right? The same here principle. So these primer lawn are complementary to the DNA adapters. So when you introduce the single-stranded DNA after denaturing with the sodium hydroxide mediated denaturation of the DNA library produced in the first step of the sequencing, Illumina sequencing, and those DNA, single-stranded DNA, will go and hybridize to this primer lawn based on its complementarity, and then allowing the primer extension by polymerase, DNA polymerase. Okay, so after that, what you are going to do? We are going to add DNTPs and DNA polymerase so that it will go and get extend the so this is how um, the step by step. Okay, okay. So this DNA, um, the from the bottom, it is having the blue colored one, is the one what you introduce single stranded DNA. Here we are not showing different colors of the adapters, just a DNA fragment, whole thing we put as a blue color. And so the primer lawn, which is in a green color, which is immobilizedly present to the flow cell. And from here, whichever the blue color was synthesized on the flow cell, newly prepared one. So original template is this side, and newly synthesized is this side. Okay, that is how you produced a new DNA which is complementary to the original one. For us, doesn't matter complementary or the same. Once we sequence, we understand the sequence based on the either no, we can just make it into a reverse complement. We can understand DNA sequence, right? So the next step is what? We are discarding the original one. How to discard? Again, you introduce some chemical like formamide. Formamide will denature the DNA fragments. So these two DNA at present anneal together or hybridize together because of the hydrogen bonds. So when you inject the formamide to this flow cell through the hole, then what happens? Formamide disturb these two bands, two DNA strands, and once it disturbed, it will get denatured. Once it get denatured, you are going to add inject some more buffers, so that the original strand will be washed out, because original strand is not attached to the bottom. So original one is attached to the flow cell, just because of the hydrogen bond between the newly synthesized one and newly synthesized one. So once you disturb the hydrogen bonds, so the original one will be washed off. Then what happens? Newly synthesized only will be covalently attached to the flow cell. So this is how. See, the only newly synthesized one only present to the flow cell. And the DNA concentration we are going to inject to the at the beginning, inject to the flow cell such a way that it will not bind everywhere surroundingly. So it will bind, with some gaps. Okay, it will bind one of the DNA here, and all the surrounding primers will be LAN of the primers will be free, and another DNA bind here. So thus, so surrounding primers are free. So one only here, and surrounding few thousands are free, at least few hundreds are free. And after some time, some distance, and another DNA you can find, that DNA is entirely different from this, because we are adding all the library of the unknown DNA. Okay, so this is the one unknown DNA. After few hundreds, and there is another unknown DNA. Like that, it is distributed. If you want to see the whole no the understanding of the flow cell, how it looks like no, it is distributed among the flow cell such a way that surrounding few hundreds of the primers are free. And rarely it can happen two can be having very nearby. So those kind of things, we are not, it is not going to help us in sequencing. You will understand how, why it is. So you will understand why it is once you understand the next few steps. Okay, next is what you understand. This one is the primer, which is complementary to the one of the adapter. Okay, and once the original one attach here, which is having another adapter the other side. You understand? So here, so this is one adapter, this is another adapter, and in between the DNA sequence, all here represented as the only one color, but you will be having one adapter one side, another adapter another side. So these adapters complementary sequence is the green color one, and the other side adapter should be complementary to the other primer, isn't it? So that is how this newly synthesized adapter having some sequence which is we know known sequence, which is another adapter which is complementary to the this either orange or red color. Okay, so what happens, you will add some of the chemical ion strength such a way through the flow cell, it will makes the DNA to find and bend and align to the another adapter because of the complementarity. Understand? So that is how it go and bend to anneal to the another primer. It is called as annealing. Okay. So to amplify single copy DNA to a cluster DNA. So that is how. So once it get annealed, DNA polymerase you are going to add, and the NTPs you are going to add to the flow cell. So thus this primer and will get amplified until the bottom, until the this adapter sequence also, this primer sequence also get amplified until here. So you can see here. So until the other end, it is going to be uh, amplified. So then denature, again using formamide. So what happens here now? It is having the two DNA sequence. From the original one, it is formed two sequence. So is two are same? Actually not the same. In the sequence wise, entirely you can see a difference because this is the complementary to the original and this is a similar to the original sequence, newly synthesized. And these two are complementary to each other. So of course in the DNA when you take genomic DNA and information wise, both are linked, both are complementary to each other, but sequence wise entirely different, right? Except the adapters which are complementary to the primers. What happens? You are going to do the same thing multiple times, just like your PCR. You are going to do the multiple cycles. What is that? The next step again annealing. So what is that? It is go and anneal. You add the ionic strength such a way it will go and align its a complementary sequence. Then amplify. So multiple copies of it, the same thing it will make. So this is called as that's why it is called as bridge amplification. Okay, cycle repeat till multiple bridges are formed. Okay, so around 35 cycles, just like your PCR at 60 degrees, every cluster generated from a single DNA template. So all the surrounding DNA sequences, so made from one DNA sequence, these are all made because of the one DNA sequence. So this is called as one cluster, okay? And after few primers, there will be another cluster you can find. So that cluster is entirely different. They are generated from entirely different DNA fragment from this. So these are all actually generated. This one cluster, all this generated from one DNA sequence. And after few primers, you will get another this kind of cluster. All those cluster will be generated from the one DNA, but that cluster or that original DNA different from this particular cluster. I hope you are understanding. Okay. Now what we have in this particular cluster, though it is generated from a single original template, this DNA, this cluster having two kinds of DNA sequence. So this all the the alternative lines, orange or red color here, right, are having one kind of sequence which is uh, similar to the original DNA sequence, and the sequence which are present on the green color one is complementary to it. So sequencing wise, entirely different. Okay. So we cannot do the sequencing when it is two sets of different DNA sequence are there. So what we need to do, one of the sequence need to be removed. Okay. So again, here you try to understand so how nicely it was designed. See the orange color one and the green color one. So what are those sequence we understand? I explained to you this orange and green color, which are complementary to the adapters which you added at the beginning, right? So that means we know the sequence already. So this primer, which is immobilized on the flow cell, was designed such a way that this is a complementary to the, let's say, 5' adapter, and also it is having a small DNA sequence. Also, it is having a small DNA sequence which can be recognized by one restriction enzyme, which is recognized by one kind of restriction enzyme. So you know what is the restriction enzyme, right? Restriction enzymes, especially, of course, we are not much using type 1 and type 3. Mainly, we are talking about type 2. What it does, it will recognize a DNA sequence which is specific to that particular restriction enzyme and cut the DNA within that sequence. So we introduce one restriction enzyme here because we wanted to cut this DNA whenever we want. So what happens when we cut this DNA? All this green color is having that particular sequence. So when we introduce that particular restriction enzyme, all this green color one will get cut, and what happens? We will get only one kind of DNA in this cluster, which is either complementary DNA or original DNA sequence, any one kind of things we have. And now within this cluster, all the DNA sequences are identical. That is now a clone. One original sequence, make it into multiple copies of it. That is a clone DNA. Okay. So it is a cluster clone. Okay. Because now we are using the term as a cluster. So this is the clonal cluster. And these kind of clusters will be formed in the flow cells, few millions, not few thousands, few millions of cluster generates on the flow cell. And each cluster, this is one cluster, is having identical sequence. And each cluster is having identical, but in between the cluster sequence is not comparable, entirely different sequences you can find. Okay. Though next, what we are going to do? We are going to do blocking. So what is mean by blocking? So we are using some modification of the last nucleotide by using a terminal transferase. We are blocking it so that it will not elongate anymore. So it will not add any other new NTPs, new nucleotide. It should not be added. So we are going to add to the 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 newly synthesized DNA sequence, and at the same time to the primers, whichever the primers which you have. And some, once we cut using the restriction enzyme, not everything we are cutting, some DNA sequence still there. Okay, so this DNA sequence also need to be added the 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 blocker, so that it should not be get elongated anymore. So this is the fourth step in the cluster generation. And the final step of the cluster generation is, so again, you have this sequence, which is the adapter sequence, and other end also, right, which is complementary to the another adapter. So once we know this sequence, which is what we have added at the first step of the library preparation, that mean adapter. So we know this sequence, this end also we know, this end also So this end, once we know, it is easy for us to design another small sequence which can be used as a primer. So what we are going to do now, we are adding some small primer sequence to the flow cell, and which is complementary to this adapter. So once we add to this flow cell, which is complementary to here, automatically it will go and align here. So that means we are hybridizing the another primer to the another side of the newly synthesized, the clones, the cluster. So whole thing within the cluster, all the sequences having attached to the small DNA, which is complementary to the adapter sequence complementarily. Okay. So with this, the cluster generation step of the Illumina sequence completes. So what you got by this, you got now the clusters, if you want to have just to visualize the mind, I mean, illustration is like this. So it is having the flow cell. So clusters was generated. So this is the blue dot. Let's say this is the a full cluster. So that one cluster is having thousands of similar sequence. Okay, each dot, and this dot also thousands of similar sequence. But this sequence is different from this sequence. Sequence is different from this sequence, and like that. So each cluster is having different sequence, but within the cluster all sequences are identical. So that is how it is generates in the by the end of cluster generation. So the next step and a very important step is sequencing, where you will get the data. Until now, you didn't get any data. You're just preparing for the sequencing only. So what you will get now, you will get a real sequence of the DNA. So now the time is 10:05. So as I used to tell, I don't want to take in a stretch. What you can do, you can take 5 minutes break. We will continue. So we will continue by 10:10. So now the time is 10:04. We will start again back by 10:10. Just to relax, stretch your body, and walk a little bit. Okay. So prepare for next few more minutes of class. Okay. So I will continue in 5 minutes. Have a break. Okay. So by now what happens, we have the two steps completed. The first step is library preparation, where you are making the DNA, a long DNA, whatever the big chromosomes you have, just making into small pieces, and then adding the adapters, 5 prime one adapter and 3 prime one adapter. You are adding to the flow cell, which is having the complementary to these two adapters. So when you introduce to the library of DNA to the flow cell, it will bind to the flow cell with because of it's a complementary to the primers which are present on the flow cell. And that binding was the concentration of the DNA and the primers present was calculated such a way that each DNA randomly it should bind, the DNA fragment will bind a few hundreds or of primers afar, it should not be very near. So, but random possibilities are there, no? So there are some primers which are, there are some DNA which can bind very nearby. Those things, which will result in a different way, which I will explain at the end of the sequencing. So those sequencing results, which it will be not useful. Or any other kind of problems like some of the cluster which is not properly generated. So those also will be not much useful. So mostly, the once it get annealed, and then in each step of the cluster generation will be successful. then it will get uh, a cluster of each clusters will be generated from each individual DNA bound in the first step of the cluster generation. Okay, so then we'll go to the sequencing step. So this is how it looks like, and where in the sub steps, so if you see, you have um, incorporation, scan, and cleavage. Okay, so we'll understand those. Okay, so again you recap your knowledge on your PCR. What happens in the PCR when you add a DNA polymerase, DNA, template DNA, and primer? Everything is there. What happens? The DNA polymerase will add one more nucleotide to the primer based on the template DNA sequence, right? Which is complementary to that. It will add and add few nucleotides, keep adding. So that is the elongation, right? So that is the same thing happening here in the sequencing, because this template DNA, the elongated DNA, the newly synthesized DNA in the previous step. So in order to show again, this is the one. So the long stretch of DNA, which is newly synthesized in the cluster, all are similar. And primer, we annealed to this one of the end, which is complementary to the original DNA based on the adapter sequence which we knew already. So this is how this primer here we attached to the this DNA fragment which are present in the cluster. After this, we are going to add all the nucleotides. So in order to compare with the Illumina, we are going to add one nucleotide at a time. So just we inject first D ATPs, and then see the peak of the light, and then wash it out, and D GTP and wash it out like that. We these are the steps are, like injecting the nucleotide, visualizing the light, washing, again injecting the second nucleotide, and visualizing the light, and washing. So each time one only, it will inject in case of pyrosequencing. But here in Illumina sequencing, it is not like that. In order to understand comparatively, just to explain you. So here 4 nucleotides you are adding to this flow cell at a time, you are injecting. So this is how it is looks like. So here the flow cell is attached. You are going to inject those nucleotides and collecting all this information. I mean, sorry, the ingredients. So when you add all these 4 to the flow cell, these DNTPs are not just your normal DNTPs which you are using in PCR. These are having two modifications. These are having two modifications. What are those? One is each nucleotide is having attached to the different fluorescence. G is one fluorescent dye, C is another dye, A is another fluorescence dye, and T is another fluorescence dye. So like that fluorescence, these 4 nucleotides are attached to the 4 different fluorescence dyes. That is one thing. And the second thing is all are having blockers. So these blockers are such a way. See, let's say now this is a C. When it is a C, is a G is attached to the added to the elongated primer. Let's say this is the A or something, just to explain to you. This is the A, and supposed to be T need to be added, but T is present, but it will not get added because blocker is there. So what is the conclusion here? Though the 4 nucleotides are present, it will add only one nucleotide at a time. Okay. So even though we are injecting all 4 to the flow cell, but it is attaching to the primer only one nucleotide, which is complementary to this first nucleotide only. After that, it will stop. even though the nucleotides are there. So that is the incorporation first step. And once it get incorporated G, so this fluorescence will be, released, and it will gives the color, I mean the fluorescence. And this fluorescence will be detected by the detector, captured. So this fluorescence will be captured. So that particular cluster will be fluoresced with the G's color, maybe purple or whatever it is. It will fluoresce, and this will be detected by the detector. And then that is the scanning. It's called as scanning. But with our visual naked eye, you will not see anything. You will just see like this only. You can't see anything. Okay. Then after that, cleavage. What is the cleavage? This blocker, right, need to be removed. So there is another chemical or enzyme you will add where this blockage will be removed. So after blockage is removed, again you go for the incorporation. Incorporation means you will add ATCG all, and here A is added. So let's say this is a T, A is added. Only A is added, even though other nucleotides are there, others will be not added because of the blockage. And here the fluorescence will be released, maybe red, which is the detector will scan and capture it, and store the information as this is the red. And then after that, and again cleavage, remove the blockage. And thus you are going to do every single nucleotide, you are going to do these steps: incorporation, scanning, and cleavage. Okay, so scanning is actually how, it is like taking a photo. This is the camera. See the camera. This camera is a very, very highly uh, uh, a sensitive camera, like pixel, uh, very high pixel camera with the lenses, and then it will take this kind of picture. So once it take the picture, this is one tile. So each column, this is one column only. Okay, each column is having two rows. Sorry, each channel is having two columns inside it. Each column is having multiple tiles. See, this is a one tiles, two, three. There are so many tiles will be there, and each tile at a time it will take to capture the picture. And each the tile will be having some thousands of clusters, and it will take the picture for each nucleotide added. After each adding, it will take the picture, but it will not take one picture. It will take 4 images. So see, the each flow cell is having 8 channels. That we have understood, right? Each flow cell is having 8 channels. Each channel is having two columns. See, this is a two column. This is only one channel, but two columns are there. Each column are having 60 tiles. 60 tiles are having. Okay, and each tile it will take one picture. Each tile it will take one picture, but not one. Each time one nucleotide add, it will take 4 picture in order to take an if there is any wrong with one image, it will take an average of it. It will try to take the information from other image. So it will take 4 images. Okay, so yeah, when you are sequencing, this is the one nucleotide added. One nucleotide added, 4 images taken for that particular tile. So there are how many tiles? There are so many tiles. So 60 times 2 times 8. So 120 times 8. That means around 960 tiles will be there, whole flow cell. And each tile, it will take 4 images for each nucleotide, and it will keep on adding nucleotide and sequencing. Let's say it is 100 nucleotide sequencing. So as I told you, here there is a limitation. It is not going to sequence all thousand or 600. It can sequence around 100 only at a time. So 100 it will do, and times 2, this will, I explained to you why it is 2. It is called as paired end sequencing. And 100 times every nucleotide, 4 images for each tile. So you will get 640,000 images. Each image, as it is high pixel image, so high megapixel image, it will take 8 MB of size minimum. And for every run, it required a few terabytes of the space for the computer to be, to store this information. But of course, as a naked eye, it is not clear. But it is when you go in 100 microns zoom. So this is how it looks like. See, each cluster, within the cluster, few hundreds of sequence are there, but all hundreds of the sequence will take a particular kind of nucleotide. See, here the G was added at the first beginning. The purple color was given. So not only this particular sequence, all the sequence present in the same cluster was added G. All G's given the same fluorescence. So it will be easy. It will be easy to capture the light. So that's why. So that G, maybe let us say like a blue. Okay, here it's not purple. It's not this. Blue, green, yellow, red are there. So let's say there's a blue added. So the first nucleotide that G added, blue color emitted. So that blue color was captured by the camera, and this one will be later. Next step, the fourth step is analyzing. The fourth step, analyzing, it will, the blue color will be decoded into G automatically. Here at the same time, another cluster was taken different nucleotide, maybe C. Another cluster taken A, another cluster taken T. So at the same time, all four are possible. All four nucleotides we added. Each cluster take whatever the thing it want, either ATCG, and based on which nucleotides taken, and they releases different fluorescence, either C or T or A or G. So based on that, all four kinds of colors you can see. And at 20 microns one, you can see like this. 100 microns zoom, you can see like this. And you can there will be 50 million cluster per flow cell. Each flow cell, the which is having 8 the channels, all together you will be having 50 millions, all 50 million kinds of dots you can find. Okay. So, but reality, what you can see like this only. You cannot see much difference in the colors in the naked eye, but in the detector can do it. Okay. So that is about the actually the whole sequencing of the Illumina, including the library preparation, cluster generation, then sequencing, and analyzing. Analyzing is what? Using this data, it will be get analyzed, which color released and what would be the nucleotide. But at the same time, you can see some of the things very nearby clusters formed. For example, here see yellow and blue. Is this overlapping? That means the DNA, when it is annealing at the first step of the cluster generation, it was annealed nearby, not that far. So that's why it is overlapping. Okay, so sometimes this also can, but sometimes this, see the blue, red, blue, this kind of things are this kind of thing, when you cannot much differentiate. Much closer mean you cannot differentiate. So those kind of data, you cannot utilize it. See here, yellow and red. So this kind of data you cannot use for the sequencing. Okay, or here. So those data, it will be excluded for the sequencing information. So other sequences which are individual clusters are there, it will be taken. Maybe around 10% of the information, maybe it may be no use. I mean, based on the how the concentration exactly properly you injected. If it is more concentration of DNA introduced, the more overlappings are possible. If it is less, there is a less chance, but you will not get occupied to get 50 million. So it was standardized in such a way that the some concentration is ideal so that you will not get overlapping and still you will get the 50 millions of clusters. Okay, so by this you have sequenced one, the around the first 75 or 100 nucleotides. After that, there is a limitation; it will not get sequenced. But at present, this is when it was started in the 2010s and 11s. It was like that, but 75 nucleotides. But after that, it advancements in last few years, it can sequence until 300 nucleotides also. 300 nucleotides also you can sequence in the one stretch. But anyhow, it is having an advantage of the Illumina. You can do the paired end. That means one end you sequenced first 300 nucleotides. In another end also you can sequence another 300 for the same cluster. The same cluster you can sequence without any problem. So this is the one, the DNA sequence if you see. So you have used this one, a primer, read, and you sequence one stretch of DNA. And the same DNA sequence you can sequence from the other end. Okay, so that gives more confidence on our sequencing data. How to do that? Actually, if you have understand until now, it will be almost the same to use the paired end sequencing. So what is the one we are going to do in different illustration I'm showing here. So the flow cell, when it is having the bridge amplification, so it was formed two kinds of DNA sequence in each cluster. That means each one is original, another one is complementary. When it formed one of the strand, we have cut using the restriction enzyme, and we have just left only one kind of DNA in one cluster, multiple copies of the same DNA, and we use the complementary to that one adapter and sequenced it, isn't it? So that is what we have done in the so far. So next, what we can do, we can remove the blocks what we are having at the end. Say here, um, we are having the blocks at the ends, right? So those blocks we can remove first. So unblocked, 3' ends are unblocked. And then once it get unblocked, this go and anneal to the, again, the same thing happens and amplify it. So the same thing, again, you are repeating the same thing and making the bridge amplification and make the isothermal amplification and yeah, isothermal amplification and make multiple copies of it, then again you block it. Exactly, you are repeating the same thing like cluster generation. Okay, so what we are doing, first we remove the blockage and making the multiple copies and make the two kinds of strands, which is original and complementary. Okay, after that what we are doing, we are using, some of the slide is missing, what we are doing, we are using the another restriction enzyme to cut the another strand. So we are using, let's say here the figure showing one of the strand is blue color, another one is green color. So first we have cut the blue colored one and sequenced the green color one, which is a complementary sequence. If you think, then we are amplifying again back by unblocking and making the same kind of bridge amplification. And now we are cutting the complementary sequence and making the original sequence as it is. Okay, so that means we are going to have another DNA sequence which is complementary sequence to the sequence what we sequenced. Okay, so then again we know other end also, right? So make the make the primer design complementary to this adapter and make this anneal to the this one and block it the whole thing. So that means the the DNA sequence which you need to sequence also block it, and other adapters also you just block it to not have any more bridge amplification, and go for the sequencing reaction again. So what is that? You will get you will add the all the nucleotides, and each time one nucleotide it will get amplified. Then it will release the fluorescence, and based on the which fluorescence released, and it will decode it into sequence. So what is the advantage of it? The same cluster. So the camera, the software will remember the what cluster it is, you know. So the same cluster, the first 300 you found, and the same cluster another 300 you find, and the the software can it will gives the information to you that the first 300 and the next 300 from the same cluster, this is what generated. Then when we are analyzing the data, we can do some using some other tools, the sequencing aligning tools, pairwise paired end sequencing, and it will go and anneal with the same sequence to understand whether it is having the overlap or not. So sometimes it will have huge overlap, sometimes it is having small overlap. So what is the advantage of it? When you have 300 forward and 300 reverse, the whole sequence, let's say 500, so 300 and 300, and you will get how many overlapping sequences? You will have 100 overlapping sequences, and you will get a stretch of 500 nucleotide sequence, you will get it. Okay, so that is the one advantage. And second advantage is that when you have an overlap, it will have more confidence on your sequence. Let's say only 300 nucleotide is your sequence, 300 forward you sequence and 300 reverse you sequence. Let it overlap, and when it overlap, it should have the similar sequence. Okay, if it is not having the similar sequence it results, that means one of it is having some wrong, so the confidence level will be lowered. So based on that, when you are analyzing, it will give the confidence percentage also. There are some softwares it will give the confidence percentage of your sequencing results. So that those are the advantages of paired end sequencing. Okay, so with this, the Illumina sequencing whole methodology, it's completes. So now we try to understand what is the applications. So applications, as I used to say, is a huge applications. It is whole genome sequencing applications and a targeted genome. So it no need to be only a whole genome. Like when you take a human genome, whole genome, you need to be sequenced. Or targeted genome mean, okay, if you have an idea of one person is having some mutation in 11th chromosome, so just take a 11th chromosome and sequence it. Or somebody is having some kind of some area of chromatin is having an error, you want to sequence those, so you pull those chromatin by targeted, and those chromatin you can sequence. A long PCR, so you have a just not the whole chromosome or no whole genome, just a long PCR like 200,000 or 10,000 fragment you have, you can sequence it. And epigenome, So if you want to have any kind of modifications of the DNA, some modifications, right? So like methylation kind of modifications, whatever the modification where occurred. So methylation modification information very important to understand what are the genes are silenced, got silenced, and what are the genes are actually is there the active. So by just knowing the gene is present in the genome, that doesn't mean that that gene is expressing in that particular patient. So maybe got just sequence silenced due to some error. So we need to, if you want to understand whether epigenetic modifications also, there are some ways, you can pull those the modified areas and then go and sequence it. And whole transcriptome sequencing. So these are all genome. So then to the transcriptome, as I told you in the first class in the basics of transcriptome is, what is a transcriptome? Whole RNA which transcribed inside the cell. Okay, it is not only the mRNA, all the RNAs. Whole transcriptome at that particular moment, at particular cell, what is their whole RNA, that is a transcriptome. So if you want to have the the liver transcriptome, that means the liver cells you take and some of the cells you take and transcriptome whole the total RNA you collect, so that is you sequence that, that is a liver transcriptome. Okay. Or if you want to understand the different developmental biology, so if you want to take, you take a fetal cell or infant cell, and you want to compare to the adult cell, the same origin, like liver only, but at different stages. And that transcriptome is different from the different stages. So that is how you can understand the developmental biology. That is a total transcriptome. That is all RNA. Or you can take only some RNA, poly A, mean that is only mRNAs. mRNAs all will be having the polyadenylated or the 3' end. So you pull down only the polyadenylated the sequences by pulling with the oligo DT. There are some technologies are there, oligo DT technology. So you can only capture the polyadenylated sequences. That means only mRNAs you can collect, and you can exclude other. Only you collect the mRNAs and you can sequence mRNA, or only you can collect a 9-coding RNA or small RNA. So many different kinds, or protein DNA, protein RNA. What does it mean? So you have some protein which will be binding to some DNA, and you want to know what are the DNA binding to that particular protein, or RNA, which are the RNA binding to that particular protein. Uh, so you pull, so you can make the protein, purify it, you bind all the RNA to it, and unbound RNA you remove it, and bound RNA you collect it and sequence it. Thus you will understand what are the RNAs binding to that particular protein, to understand the role of that protein, and also understand the functions of those RNA. So that is the, also you can do actually in our laboratory. And our students, my PhD students and master students, though my much major research is molecular biology, but we cannot exclude the sequencing technology in our molecular biology. So they are working in RNA protein binding, so they are binding one of the protein which is having the function in the pathogenicity of the some of the microorganisms, pathogenic microorganisms. So what they are doing, they are purifying that protein. They are cloning the all genetic engineering, right? They are using all genetic engineering tools to clone, and they are using all downstream processing to technology to purify the protein. After purifying the protein, they are binding to the RNA. After binding the RNA, we are sequencing using the Illumina. After that, we are analyzing the data. So the analyzing the data we are not covering in this bioinformatics, because that itself a different course that we are covering in the next generation sequencing course. We are only covering in the bioinformatics students. If any one of you are interested, even with your curiosity, once you come back, you let us know. We can also show you how it works. And yeah, we are even sequencing the total transcriptome. Also, we are sequencing the whole genome in our laboratory. We are analyzing. We are not sequencing in the sense we are sending the sequence. Once we collect, we are sending the sequence for the outsourcing, and then we are getting the crude data. We are not asking anyone to analyze. Okay, there is also outsourcing is there. So if you are not okay, you are not a really bioinformatician, you are not understand, you didn't learn next generation sequencing analysis, so you no need to worry. There is that is also there are some companies, just you give the crude data, they will analyze whatever the way you want, and you will get this the data in a very nice graphs and all. So that is not an issue. But what we are doing, we are not outsourcing that. We are getting the crude data. We are analyzing, our student analyzing by our own, because our analysis is entirely different from what they are offering. We have our own pipeline to analyze. So apart from the normal availability, we standardized our own way. So we are analyzing. So which is very interesting. We are finding because of that, we are finding very interesting data. So that is one of the example I want to give you. The sequencing is giving a lot of information which we didn't know before. So last few years only this actually this developed, and discovering a lot of information, and the information is very huge, because of this information, this sequencing, we are getting GB of data. When we are getting GB of data and we are analyzing in one aspect, see, every researcher are sequencing the particular kind, any of these area, any of these molecules, sequencing to understand, okay, their idea is whether this bind or not, this particular RNA is bind or not, this particular DNA is bind or not, whether what are the sequence present in that particular cell or particular age, only that information they are taking. But there are so much of information in that. Okay, so that information, no one is, anyone can study. So some of, you know, for example, okay, I'll explain that one. So these are the different kinds of units from Illumina. So HiSec 2000, GA2X, there are so many. These are few, and these are all like the GB per run how much, and GB per day how much, read length how much. So at that moment is this was I prepared these slides few, couple of years back. Even though I was updating time to time, but this couple of years I didn't update. This one is at that moment only 2 times 100 only, and now it is can go 2 times 300. Apart from that, almost same. That's why I didn't update. So 2 times 300 also you can go. That means 300 forward, 300 reverse. You can sequence each DNA. Can you imagine how much is efficient now? So of course there are some pros and cons are there. Even the Illumina. So the advantage are no more of cloning steps which are needed in the gunshot sequencing in the which we have used in the human genome sequencing using Sangers. You no need of that. That is very laborious. And from purified DNA to sequencing, directly you can do, and fit the laboratory benchtop, small good space accuracy. So if you have money, so even any lab can start this one, not too much of space required. Because of this is having multiplexing and paired end. So you no need to worry. In the Sangers, what you can do, even though it is automated, 96 well played, 96 sequence only you can parallelly sequence after automated. But here you can sequence millions of it at a time, and paired end also. The Sangers, what you have to do, you have to sequence forward one reaction, reverse one reaction separately, and sequence separately. But here no, paired end at a same flow cell you can do the paired end. approximately 2000 euros per GB. That was, yeah, this is another difference. So one of the things which need to be updated is one is here, 2 times 300 you can go. Another one is cost still got reduced. Previously it is 2000 euros, which is very very low per GB cost compared to the Sangers. Sangers, it required a lot of, even now, whatever the automated, it requires a lot of cost. But 2000 euros per GB a few years back, now I think even 50 euros also enough, I guess. Not even 2000, 50 euros per GB also fine. Okay. Uh, yeah, well fitted to different kinds of modified sequencing. And cons are what mission is very expensive. If you want to, sequencing per cost is low, but establishing is very high. You cannot just every lab can buy and maintain. So the mission itself very cost, reagent is cost. If you have any maintenance problem, that is also very expensive. And main error type is mismatch. There is a possibility, mismatches are there. That's why you need to repeatedly do. That means the if your target genome or gene is, let's say, 10,000, so your sequencing is not just fitted to the 10,000. You have to have multiple of the small fragments in order to make each nucleotide multiple times need to be get overlapped, so that you can reduce the mismatches. Read lengths are still too short. This also not really need to be modified, not too, still short, I can say, because 300 you can get, but Sangers, if you compare Sangers until 1000, it can go up to, but still still short. Poor coverage of AT rich region. So if you have too much of A's and T's or the same region, sometimes it is having some problem. Then, yeah, this is two actual advantages. Most widely used NGS platform is Illumina, and requires least DNA. You don't need to have too many micrograms of DNA. A small amount of nanograms also, you can sequence it. So, yeah, this one information I have taken from the Illumina website. And because when I was started teaching this, nowhere I can find those information, none of the textbooks, nowhere I can find, even videos also not there. So I have taken from that slide, and I went those workshops and got the information and started teaching. And yeah, this also applications of these NGS is metagenomics, genomics, transcriptomics, proteomics. So once we started, actually, once we understand and started uh, the sequencing technology, not only teaching, we also adapted in our research. So now most of our research having the transcriptomics, genomics, metagenomics using next generation sequencing. And not only that, we are not even depending on any others. We are doing our own analysis. So that is, when you do own analysis, you will have lot of advantages to analyze your own way. Okay, and yeah, this is one of the the immediate. So when they started in 2011, the sequencing technology, in even 2011 itself, one paper published in genetics in medicine. So this one of the girl was having some uh, the genetic mutation, deleterious genetic mutations, and due to that having some fatal disease disorder. So after sequencing, they found where the the they diagnosed what is the problem, and based on that, I think they have taken some steps to how they can. It's not cure, at least some of the steps to reduce the the symptoms of that. So this is one of the immediate first uh, the output, what do you call, paper published for the application of Illumina sequences and saved the a child life. If you are interested, you can read this paper. And this is the comparison with the existing NGS technologies. What we learned now is 454 pyrosequencing and Solexa, which is Illumina sequencing, and which is taking emulsion PCR as a feature here, sequencing by polymerase, and here bridge amplification and polymerase, and solid and pollinator also there. They take emulsion PCR as the cluster generations, and ligase as a sequencing method. Heliscope is a single molecule, also enough. This is another big advantage. And polymerase is the sequencing. And cost-wise, it's each times is varies. And but is Solexa is I mean still is a competitive, is a very low per megabase page, $2 per MB. So this is the information I want to show. When the Sanger sequencing was there, only there until 2006, okay, and the information, sequencing information was like this, it was flowing, okay, and in NCBI is collecting all the information, sequencing information from the Sanger sequencing. When the massive parallel sequencing came, like pyro sequencing, it's 2006 somewhere it started, and in 2011 or 12, it was like this. It is a huge amount of data is getting. We are getting GB of data because of the pyro sequencing and the early Illumina sequencing. And now advanced Illumina sequencing and heliscope sequencing, all those things, the the information what we are getting this much, the deluge is overflowing, and and analyzing techniques are limited, and analyzing people are now limited. Of course, there are so many people trying to use the artificial intelligence to analyze the whole data, but still there are some of the data is not fully analyzed. That's why there are so much of need of bioinformaticians throughout the world. Of course, there are some limitations in a Malaysian point of view, because not much bioinformatic based laboratories or industries established, but still there many of the labs are required by informaticians. But in the worldwide there is a huge vacuum of bioinformaticians. And yeah, because sequencing information is the deluge because of the NGS. This is throwing of the data from the thing. So global shift to sequencing based technologies. So previously they used to understand many of the uh, unanswered questions by proteomics, microarrays, these, that, still they are doing, but wherever the answers can get by sequencing, everyone going for the sequencing. They are, when whichever the questions not answered by the sequencing technologies, only they are going for microarrays and proteomics, because the advancement is one-sided. There's a huge development was occurred in the last few years in the NGS, and it is it taken uh, full advantage of it. And that's why we are many of the area of research, first they are trying to understand the unknown information through the NGS, so, or at least preliminary information to understand, then they are going for secondary validation methods. Okay, so this is about the Illumina sequencing, and thank you everyone.